Hello everyone, I'm Hyungsoo Jeon and I'm in Department of Biological Sciences. Following Titinam Swanan's presentation, today I'm gonna talk about the latter half of Chapter 9, Interleukins and Tumor Necrosis Factor. I will present Chapter 9 with this content. First of all, I will introduce about Interleukin 1 and 11, next tumor necrosis factor in detail, and wrap up with summary. Let's begin with Interleukin 1. Interleukin 1 is known as lymphocyte activating factor. It offers wide range of biological activities and has been appraised clinically in several trials. There are two forms of IL-1, IL-1 alpha and IL-1 beta. Although these two forms of IL-1 exhibit only 20% homology, they bind the same receptor and induce similar biological activities. They are originally expressed as precursor protein, and the major protein is released by proteolytic cleavage. Both IL-1 has similar globular beta-beta structure. It is composed of 6 strands of beta sheets. Lastly, IL-1 is produced by a wide range of cells. Among them, macrophage is the main source. The major function of IL-1 is to induce pro-inflammatory effect. IL-1 promotes the synthesis of various inducers of inflammatory mediator, such as eicosanoid, and at low concentration, it induces local inflammation in a paracrine manner. In addition to inflammatory role, IL-1 activates lymphocytes, and at high concentrations, it causes hepatic synthesis of acute phase protein, fever, and cachexia. In addition, IL-1 promotes hematopoietic cell growth and differentiation. There are two clinical applications of IL-1. Initial clinical trial of IL-1 was focused on immune stimulatory effect that restores hematopoietic processing during chemotherapy and shows anti-tumor effect, but the result proved disappointing. No significant anti-tumor response was observed in many cases, although side effects are commonly observed with limited dose. Another approach is IL-1 signal inhibition to reduce inflammation and local damage. Among several antagonists, recently a native IL-1 receptor antagonist has approved for RA therapy. Its trade name is Kineret. Next, I'm gonna briefly talk about Interleukin 11. IL-11 is 20-30 kilodalton cytokine produced largely by IL-1 activated bone marrow stromal cell and fibroblast, and acts as hematopoietic growth factor. It stimulates thrombopoiesis, that is platelet synthesis, and growth and differentiation of bone marrow cells. The platelet synthesis stimulatory function is used for clinical application. Numega is the IL-11 based product approved for prevention of thrombocytopenia that is caused by low blood platelet count of cancer patients. The product is produced in engineered E. coli cells and is applied in freeze-dried format. Lastly, I'm gonna introduce tumor necrosis factor. Tumor necrosis factor family of cytokines consists of two related regulatory factors. One is TNF-alpha and the other one is TNF-beta. Like as IL-1, two different forms of TNF bind the same receptor and has similar biological activity. In most cases, TNF-alpha is referred to simply as TNF because TNF-alpha has been studied in significantly greater detail than TNF-beta. Human TNF-alpha is initially synthesized as a precursor protein that is anchored in the plasma membrane by a single membrane spanning sequence. This TNF precursor protein itself displays biological activity it is usually proteolytically processed by a specific extracellular metalloprotease, and this proteolytic cleavage occurs between residue 76 alanine and 77 valine, yielding the mature soluble TNF-alpha. Monomer TNF is biologically inactive. The active form is a homotrimer in which the three monomers associate non-covalently at about threefold axis symmetry, forming a compact bell-shaped structure. Also, macrophage is a main cellular source of TNF-alpha. And there are two TNF receptor types have been characterized: TNFR55, TNFR75. It appears that TNFR55 is capable of mediating most TNF activities, whereas the biological activities induced by the TNFR75 receptor are limited. This phenomena is in accordance with their expression patterns. Although all cells express TNFR55, the TNFR75 cell surface receptor distribution is more restricted. The binding of TNF to either receptor type results in oligomerization of the receptor. This oligomerization of the receptors is often followed by their phosphorylation of their intracellular domains. Following clustering of the TNF receptors, these phosphoproteins are likely to become activated, thus mediating additional downstream events that eventually trigger characteristic TNF molecular responses. Tumor necrosis factor alpha is a pleiotropic inflammatory cytokine involved in diverse cellular responses. The major biological responses are as following. Their specific and non-specific immune response is particularly caused by gram-negative bacterial infection. TNF-alpha also induces inflammation and presents relative cytotoxicity against some tumor cells. Let's see in detail. At low concentration, TNF-alpha activates both specific and non-specific immunity. These TNF-alpha actions include activation of various phagocytic cells and enhanced toxicity toward pathogens. It has antiviral activity, increased cell surface expression of class 1 MHC molecules on sensitive cells, and enhanced proliferation of IL-2-dependent T lymphocytes. TNF-alpha also plays a prominent role in mediating the inflammatory response. Indeed, this may be its major normal physiological role. It promotes inflammation by a number of means. First of all, it promotes activation of inflammatory leukocytes and enhances expression of various adhesion molecules on the surface of vascular endocellular cells for docking site of various inflammatory cells. Also, it displays chemotactic effects and enhanced vascular leakiness. Furthermore, it promotes synthesis of various lipid-based inflammatory mediators and pro-inflammatory cytokines. Initially, people are interested in utilizing TNF-alpha as a general anti-cancer agent. Veromun is firstly approved as anti-cancer agent. This recombinant product is identical to the native human TNF-alpha, but their general anti-tumor effect has diminished, largely due to the realization that, firstly, many tumors are not susceptible to destruction mediated by TNF-alpha, and secondly, severe side effects usually accompany systemic administration of therapeutically effective dose. As a result, this agent used only for local perfusion against some specific tumor like as melanoma. So most clinical interest in now has focused on neutralizing its biological effect in situations where overexpression of TNF induces negative clinical effect like as RA. Enbrel is a TNFR75 and human Fc fragment fusion protein product approved for medical use that is based on this strategy. Enbrel functions as competitive inhibitor of TNF-alpha. Binding of TNF to Enbrel prevents it from binding to cell surface receptors. Also, the antibody Fc component of the fusion protein present extends serum half-life on the product, increasing it by 5-fold relative to the soluble TNF receptor alone. To sum up, I have introduced three kinds of cytokines: IL-1, IL-11, and TNF. These cytokines are naturally produced in our body and closely involved in immune systems. Also, these cytokines are easily expressed by recombinant technique to utilize for therapeutic application. Generally, these cytokines are very toxic to be used systemically, so the potential side effects should be considered. In this sense, anti-IL-1 and anti-TNF therapy is more effective in clinical use than IL-1 and TNF therapy.